You've seen the headlines. A food product has been recalled. No illnesses have been linked, but the company that manufactures the food found a dangerous pathogen during routine testing. But the headlines change. Now there are reports of illnesses, all from the same food. How are you affected? Did you find recalled food in your kitchen? Do you know someone who has gotten sick? How did the food company determine its product was contaminated? Who figured out that people in different states and even different countries became sick from the same food? Let's take a step back in time and learn what came before the headlines. When certain types or numbers of illnesses are reported by physicians, investigators look for possible connections. One connection could be the patients consumed the same contaminated food. Pathogenic organisms can contaminate foods before harvest, during processing, and during handling. Pathogens are commonly transmitted through the fecal-oral route and can persist in the environment. Public health departments and regulatory agencies investigate potential foodborne illnesses. Epidemiologists interview patients to determine if they consumed something in common. They may ask what and where a person ate for the last 3 days or even 3 weeks. Imagine trying to remember everything you ate and drank that long ago. Human specimens and samples of food suspected to be linked with illness are collected and sent to a laboratory. Welcome to the lab. Kirsten, our microbiologist, will try to determine what, if any, pathogens are in these samples. The patients report symptoms of acute gastroenteritis. Many types of microorganisms can cause these symptoms, including bacteria, viruses, and protozoa. So where do we begin our lab investigation? We begin with an understanding of microbial structures and behaviors. Let's first consider how these organisms are alike. They have genetic material at the core. They are also made of proteins, lipids, and carbohydrates. But they differ in specific molecular composition and function. We use these differences to detect and identify them. Viruses are the most structurally simple. Some viruses have DNA, while others have only RNA. They have a protein coat called a capsid. Some also have an envelope. Though known foodborne human enteric viruses are non-enveloped. Viruses do not take up nutrients, create energy, or replicate outside a host cell. Viruses won't increase in number in the food, but they can survive in foods. Viruses are shed during illness and can persist in the environment. Protozoa associated with foods are similar to viruses in that they do not replicate outside a host, though they persist in the environment. The transmissive stage is an oocyst. Its genetic material is protected from environmental stresses by many outer layers. Each oocyst may contain several infectious particles. Many oocysts can be shed during illness. Some protozoa have very complex life cycles involving many stages or even multiple hosts. Bacteria have the structures and functions necessary to replicate in food and the environment. The membrane and outer wall are made of protein, lipid, and carbohydrate. Bacteria move and attach to surfaces using flagella and pili. Surface molecules help them adhere to food, a cutting board, or to cells in the body. These are some basics. Methods used to identify microbes are based on biochemistry, including structures, functions, infectivity, and metabolism. Knowledge of microbial biochemistry is constantly advancing as are technologies used to study microbes. Let's look at a few strategies that can be used to solve the outbreak. Remember, we are trying to find microbes 1,000 or even million times smaller than a pencil point. And we can't afford to miss any because sometimes even small numbers are enough to make a person ill. Kirsten will need the right tools to determine what organism caused the illnesses and its source. Let's take a look in the lab toolbox and learn how to characterize microbes. proper handling of samples is critical. The samples will be handled aseptically, kept in a clean environment at all times and touched only by sterile supplies. Foods and human specimens are complex. Some constituents can interfere with tests working properly. Texture can complicate recovery of a pathogen. If our sample is a solid, we mix it with liquid that favors the recovery of microbes. If it is a liquid, we work with it directly. We try to grow bacteria from our outbreak samples because we have a better chance of detecting many rather than just a few. To culture bacteria, we make media that contains the nutrients best for their growth and transfer some of our sample to the media. These are incubated in the conditions best for growth and replication. This process usually takes a day. Bacterial growth appears as a cloud in liquid or a colony on a plate. We may now have large numbers of different types of bacteria isolated in pure cultures. To figure out what types of bacteria we have, we use selective media that gives us an indication of bacterial identity according to metabolic characteristics. Bacteria may utilize certain nutrients and release byproducts that can be seen by color change. But even on selective media, two different types of bacteria can look the same. We need additional tests to confirm if we have actually recovered a pathogen.
We look the same on agar and under the microscope. One of us is harmless and one of us is pathogenic. How else might you tell us apart?
I have antigens on my flagella and cell surface that are different from that one's and can be detected by immunoassay.
Immunoassays are tests used to detect and identify microbes by reaction of their antigens with specific antibodies. Antibodies are molecules produced by an immune system as one type of defense to a foreign molecule. While we are not measuring an immune response in this test, we can use these molecules to identify our isolates. We mix our isolate with antibodies known to interact with antigens on the pathogen for which we are testing. If the pathogen is present, its antigens will interact with the antibodies. And this can be visualized by agglutination or color changes. We have cultured bacteria and confirmed their identity by immunoassay. But what about protozoa and viruses? Since they don't grow and multiply in food, we must use different strategies to recover them from our samples. One method to recover protozoa and viruses is to concentrate and then separate them from the rest of the sample. To do this, we transfer our sample to a tube and place it in a centrifuge. A high force of centrifugation pulls the dense particles to the bottom, while less dense particles stay in top layers. We can separate our pathogens from the sample by differences in density. Another way to recover protozoa, as well as bacteria, is to capture them with immunomagnetic beads. Antigens on a pathogen react specifically with antibodies coated onto a magnetic bead. Beads are mixed with the sample and recovered with a magnet. We may have concentrated or captured protozoa and viruses, but we still don't know if they could have caused these illnesses. The infectivity of viruses and parasites can sometimes be tested in the lab, not with an animal model, but with just one type of animal cell. Mammalian cells can be grown in a thin layer in a flask or on a plate with a technique called cell culture. We add our sample isolate to the cells. If infectious viruses are in our samples, they invade the cells, replicate, and release progeny that infect neighboring cells. Viruses depend on the host cell machinery to replicate. Protozoa may be able to infect a cell and complete some, but not always all, of their life stages. When enough cells have been infected, cell death or other changes can be seen with microscopy. We may also stain the cells to visualize infection with the naked eye. We have concentrated and cultured our samples, used microscopy and immunoassays. These classical approaches are valuable, but can take days to complete. Meanwhile, we don't know what is causing illness or what action to take to prevent new illnesses. In outbreak investigations, we rely heavily on gene-based tests for pathogen detection. We can look for specific gene segments or look for differences throughout whole genomes. In the first approach, we isolate a unique gene segment of interest and make enough copies to be able to measure it. After initial steps to recover whole pathogens, we isolate all of the nucleic acid present by treating the sample with chemicals to degrade other macromolecules. We now look for the specific segment in the mix of nucleic acid. A lab technique known as polymerase chain reaction is used to make copies of this segment. The sample is heated to separate DNA. We add small strands of nucleotides to anneal to the ends of the segment of interest. A polymerase enzyme helps extend the complementary strands. Repeated cycles make enough copies of the segment that we can measure its presence. We detect our PCR product by measuring chemicals that react with DNA. In one approach, we prepare a gel and fill its wells with our PCR products mixed with a dye. The gel is exposed to a current and negatively charged DNA migrates through the gel. Smaller molecules migrate more easily, so we can separate a mix of molecules based on size. When the gel is exposed to UV light, pieces of DNA are seen as bands. We compare the bands to segments of known sizes. If the unique segment from a pathogen was present in the sample, it should be amplified by PCR and measured to the expected size. Another approach to detect and compare sample isolates is to look for not just one gene segment, but for differences throughout whole genomes. In this approach, DNA is cut by restriction enzymes at specific stretches of nucleic acids. The cut location varies among organisms, so the number and sizes of DNA fragments also vary. We compare banding patterns among sample isolates to help determine which outbreak samples are connected. We attempted to recover pathogens from samples and characterize them by metabolic, immune, infectivity, and gene-based assays. Lab studies are complete. These are just some of the technologies available to characterize microbes. Certain communicable diseases are reported by public health agencies to a network. The Centers for Disease Control and Prevention monitor the reports and look for indications of an outbreak. Food regulatory agencies are also associated with the network to aid communication among those who work to control the outbreak. The monitoring of significant diseases is a nationwide and worldwide effort. Now we know the cause of illness, which illnesses are linked, and the implicated food. Thankfully, the cause of this outbreak could be determined and no serious illnesses or deaths were reported. Unfortunately, this is not always the outcome. Now the headlines break and the story continues to evolve. What do you think should be done next?